Hi, my name is Michael Fujarski. I'm a research associate at the Institute of Medical Informatics in Münster, Germany. And I'm going to present to you our evaluation of mutational effects on protein structures with Alphafold. But first of all, I'd like to introduce the topic briefly. Proteins are involved in many biological processes like cell reproduction or gene expression. And those interactions with other molecules highly depend on the structure. But determining the structure is very time-consuming and therefore expensive. So it's not really feasible to determine for a set of unknown mutations whether or not they do have an impact on the structure. In order to minimize the set of possible mutations, we need in silico approaches to approximate the protein structure for a mutation and create, by doing so, a subset of candidates that can be further evaluated with experimental efforts. So in order to approximate the protein structure, we picked Alphafold 2.0 since it outperformed all other participating models in the Cas14 competition. We used that for further evaluation and used it on the hemoglobin beta subunit as a benchmark. So we predicted the actual wild type of HBB as well as the sickle cell variant, a point mutation at position 7, and various artificial substitutions close to a binding site. We substituted consecutively up to 15 positions with glutamic acid or with proline. The predicted structures were then aligned using the RD kit, and the difference between two structures was calculated with the root mean square deviation, RMSD, on the C-alpha backbone. So in order to evaluate the prediction of the wild type as well as the sickle cell variant and the artificial substitutions, we also need a reference wild type structure as well as a reference sickle cell variant structure. Those were obtained from UniProt and compared with all other predictions. First of all, the reference wild type and the reference sickle cell variant were also compared in order to have a framed reference for the RMSD. So those had an RMSD of about 1 angstrom. The wild type HBB, as seen on the right figure, had an RMSD compared to the reference wild type of 0.54. And the predicted wild type and the predicted sickle cell highlighted in the table had an RMSD of 0.039. And on the right side, the substitution of 15 consecutive amino acids with glutamic acid, highlighted in red in the structure on the right side, had an RMSD of 0.85 angstrom. So to sum up the table one, the reference wild type and the reference sickle cell had an RMSD of about 1 angstrom. The predicted wild type and the predicted sickle cell had an RMSD as little as 0.039 and 15 substitutions, so about 10% of the protein were replaced, had an RMSD of 0.85 compared to the predicted wild type. So apparently, even large mutations have no effect on the general structure. But since glutamic acid has a high probability for alpha helices, we also repeated the same substitutions with proline, which is known as a alpha helix breaker. And the overall C-alpha RMSD, as seen in the bottom right figure, is about 3.14 angstrom. So it has a quite large effect on the substituted chain. But excluding those amino acids and only considering the rest of the molecule in blue and green, it still had an RMSD of 0.53 angstrom. So Alphafold does not appear to be suitable to predict any mutational effects on global structures. So thanks for listening, and if you got any questions, feel free to ask.